My name is Lars Bode. I'm a professor of pediatrics and also a director of the Mother Milk Infant Center of Research Excellence here at UC San Diego and director of the new Human Milk Institute also at UC San Diego. And to introduce MPRINT, UC San Diego is one of the two centers of excellence in therapeutics as part of the MPRINT network. And specifically, our center in San Diego has a strong focus on human milk, and hence the focus today also on human milk and how we can learn from human milk to develop new therapeutics, potentially diagnostics, and in this case, in the context of necrotizing enterocolitis, or short NEC. So I'll be talking mostly about human milk for the first, let's say 20 minutes, maybe 15, 20 minutes. What is in human milk, lead into human milk oligosaccharides, which is one of the focus areas of our research in my lab. And then slowly move into necrotizing enterocolitis. What is it? Why are we looking to human milk when we talk about NEC? And show you a few of our discoveries that now lead to developing new therapeutics and a diagnostic that will hopefully help reduce the numbers of infants that suffer and die from necrotizing enterocolitis. So let's get started talking about human milk. And I show this in many of my presentations, independent of the topic, really, to highlight the power of human milk. This is a statement from the Lancet breastfeeding series that came out 2016. And some of you might have seen that there was just a new breastfeeding series that was published in the Lancet just last week. The numbers are very similar still, and the focus of the last 2023 breastfeeding series is slightly different. So I'll keep this statement in here for now. And the statement reads as simple and powerful as this, that the death of 823,000 children and 20,000 mothers each year could be averted through universal breastfeeding, along with economic savings of $300 billion US. And we really set out to say, okay, well, why is that? You know, what is it in human milk that makes it so powerful? How does it save lives? And how does this really work on a mechanistic level? And if we know how this works on a mechanistic, potentially molecular level, can we leverage that knowledge to then develop new therapeutics and find solutions to really help all infants and potentially also save maternal lives? And just to give you a few examples here, in the infant space, we see that still we lose about 2,200 children under the age of 5 years to diarrhea every single day. And on the other hand, we know that that children who are breastfed have a substantially lower risk of suffering and dying from infectious diarrhea as well as respiratory infections. And the question is, why is that? You know, what is in human milk that saves us from these infectious diseases? In the preterm infant space, and this is really relevant to today's talk, we see that about 5% of all very low birth weight infants still develop an often deadly intestinal disorder called necrotizing enterocolitis. And the consequences are quite severe if you survive this disease. And on the other hand, again, we see that preterm infants who receive human milk are at a 6 to 10-fold lower risk to develop NEC. And the question is, well, how does this work? What is in human milk that protects the preterm infant from this devastating disease? And if we identify how this works, can we use that knowledge to develop new therapeutics to prevent NEC in all babies? And that's really the focus of this talk. But before we get there, let's take a look at what else human milk is doing. We know that the benefits of human milk and breastfeeding extend beyond the actual exposure to human milk. So it's not just benefits during the breastfeeding period, it is benefits for life. And we see that infants, children that have received human milk have a lower risk to develop overweight and obesity, have a lower risk to develop diabetes, and are potentially smarter. And wouldn't it be great to know how all that works? You know, what is in human milk in the process of breastfeeding that potentially makes us smarter, but also protects us from many of these diseases? And on the other side, we're always focusing on the infant when it comes to breastfeeding, but really we should focus on the mom here as well. There are benefits to maternal health here as well if a mom breastfeeds and the longer she breastfeeds. So for example, there's a lower risk of diabetes associated with breastfeeding, a lower risk of cardiovascular disease, one of the major killers still in the Western world, and a lower risk of breast and ovarian cancer. So again, knowing how this all works would greatly benefit us developing new preventative methods and measures to help women that cannot breastfeed, for example. Okay, so what is in human milk that makes it so powerful? We're always talking about the process of breastfeeding, but really, what are the components of human milk that may potentially contribute to that? Of course, there's water. Milk is mostly made of water, but then this water contains lots of carbohydrates, mainly lactose, but also human milk oligosaccharides that we'll be talking about in a minute. Then there's lipids, highly concentrated lipids in the specific form of the milk fat globule, with a membrane layer around the triglyceride core, and that membrane layer has integrated proteins and glycoproteins and glycolipids and many other bioactive molecules in itself. Then, of course, there are lots of proteins in human milk, peptides, and some of these proteins have protease activity themselves that then help digest milk proteins along the way, together with the proteases that come then with the digestive enzymes of the child. And then there's a bunch of other things. We know that there are human cells in human milk, bacteria, viruses, vitamins and minerals, of course, hormones, and a bunch of other things. So human milk is really loaded with a bunch of nutritive components, but also bioactive components. And really, human milk is more than a list of just ingredients. It's not that you have this ingredient list like you would have that on your food in the fridge. But these ingredients are highly dynamic, highly bioactive, and they depend on the context that they live in. So to exemplify that, and we'll publish this year in a perspective paper in Science in 2020, a week before the pandemic started, where you see that milk is really not standing in isolation. It is in this context of the mother milk infant triad, where on the maternal side, you have some fixed and modifiable factors that determine the different axes of this triad. Same is true on the infant side. And all this interaction is dynamic over time. We know that milk composition, for example, changes, of course, over the course of lactation, so between birth and, let's say, 2 years of age. But they really change within the day, which within a single feed, fore milk and hind milk are different in composition. And that all plays into this dynamic triad. And this triad also does not live in isolation. It lives in a context. And you can imagine that our preterm infant in a NICU here in the US lives in a very different environment as a full-term baby somewhere in a rural community in Africa, Southeast Asia, or South America. So you can see how this triad is not only dynamic, but sits in a certain context, and that context determines outcomes and interactions of that triad as well. Okay, but let's focus just on the primary macromolecules in human milk before it gets too complicated and really lead into oligosaccharides that will be the topic of the rest of the talk here. So human milk contains proteins, fat, and the milk sugar lactose, but it also contains oligosaccharides at the extremely high concentration, 5 to 15 grams per liter, which often exceeds the amount of total protein in human milk. But on the other hand, if you compare that with bovine milk or milk of other farm animals, the concentration of these oligosaccharides are extremely low, 100 to 1,000-fold lower than they are in human milk. And since bovine milk and some other milks are the basis for most infant formula, we see, at least in the past, that there were hardly any of these oligosaccharides present in infant formula. Huge contrast to human milk, where we have it really loaded with these complex sugars. I say complex sugars. What are these oligosaccharides? What do they look like structurally? They are sugars, and these sugars are made of 5 building blocks, 5 monosaccharides. And those monosaccharides are glucose, galactose, N-acetylglucosamine, fucose, and N-acetylneurominic acid, or short sialic acid. And depending on how we put those 5 building blocks together, we get different oligosaccharides. And I often compare this with Lego bricks, that imagine you had a given set of Lego bricks, different colors or different shapes, and you could now put them together in different ways, and you can use those monosaccharide building blocks or Lego bricks to build a little car, or you could take the same building blocks and build a little plane. And you could probably agree that cars and planes do very different things, and the same is true for oligosaccharides. So depending on how we put those monosaccharide building blocks together, we get different oligosaccharides, and those different oligosaccharides have different function. Structure determines function, and that's very important for the rest of the talk. Just because we call it an oligosaccharide doesn't mean that they all do the same thing. Okay, so how do these oligosaccharides look like? All oligosaccharides in human milk contain lactose at the reducing end. That's glucose and galactose. And then they get further extended by disaccharide units at first in different linkages. They can be branched, further extended, so you get a fairly complex backbone. And then you put modifications on those linear or branch structures. This group of oligosaccharides that only contain these three building blocks, so glucose, galactose, and N-acetylglucosamine, are the neutral non-fucosylated oligosaccharides. Non-fucosylated because they don't contain fucose, which is what we can add in the next step here. So we can add fucose in different linkages, either to lactose alone, and then we get 2 and 3-fucosylactose, or we add it to the growing chain in different positions, different linkages to different backbone structures. And all these oligosaccharides now that contain fucose are called the neutral or and fucosylated oligosaccharides. Now, why are they neutral? It must be something else that's not neutral then. And that's the fifth building block that comes into play here. That's sialic acid. Sialic acid contains a carboxyl group, and that introduces a negative charge to the oligosaccharides. Again, we can add it just to lactose, and we get 3 and 6-sialactose. Or we add it to the growing chain. We can add more than one sialic acid, so we get a disilated oligosaccharide, for example, down in the corner there. And all these oligosaccharides now that contain sialic acid are the acidic or silated oligosaccharides. And then we can do both. We can add fucose and sialic acid, and those would be the acidic silated or fucosylated oligosaccharides. And we don't have an example of that here on this slide. But bottom line, we have about 150 to 200 different oligosaccharides that were identified by structure. And the most important part is that not every person makes the same oligosaccharides in the same concentrations. So there is a huge variation in oligosaccharide composition from person to person, and it also changes over the course of lactation, which again plays into this dynamic interplay of milk components and then how that sits really in an environment of the triad. Okay, so research focus for human milk oligosaccharides really over the last 10 or more years has been what drives this variation on the maternal side. So why is it that some moms make these oligosaccharides and other moms make that? Can we change that potentially? Of course, most important, what are the consequences for infant health and development? What do these oligosaccharides actually do? And then can we leverage that knowledge to develop new therapeutics based on human milk oligosaccharides? And I'll do the first part here very briefly, just to give you an idea what really are the main drivers of oligosaccharide composition, and then lead into infant health and development, and then really in the end focus on necrotizing enterocolitis and what we've discovered there and how that might lead to therapeutics. Okay, so here is the variation of human milk oligosaccharides in a specific cohort here from Canada, where you see 1,200 different subjects, different oligosaccharide concentrations. Each oligosaccharide here gets a different color. And just looking at this without going into detail, you can see how different oligosaccharide compositions can be and how different they are from person to person. On the left, very low concentrations overall. On the right, extremely high concentrations. You see there's this blue color that represents 2-fucosylactose is very dominant in about 2/3 of the sample. On the right here, and on the left, you see hardly any of that color pop up on the top. And that's represented here as well. If you look at the same data as relative abundance, you see that maybe 25% or a third on the left hardly has any of that blue color. And on the right, you have a lot of it. So bottom line, huge variation between people. But if you look at the same person and you take samples over time, short period of time here, so every day on the left, so day 1, 2, 3, 4, 5, 6, 7. And then on the right, you see hourly sample taking. So baseline at 6 o'clock in the morning, 7 o'clock, 8, 9, 10, 11, 12. You see the oligosaccharide composition within the same subject is remarkably constant. And you see two different subjects here, top and bottom, but they're very different between each other. So two different subjects have very different oligosaccharide composition, but in the short period of time, their intra-individual composition is remarkably stable. And that's important because if we wanted to take a sample from a given subject, analyze oligosaccharide composition, and then associate that either with maternal drivers or infant outcomes, we know that we get a fairly representative sample if we stay within a week or two, or even over the course of a day. There's no differences between morning and evening, for example. So that's very powerful to do association studies, and you'll see that later for necrotizing enterocolitis. However, it's not that stable over a longer period of time. If we take samples from the same subject now over a course from 1 month to 24 months, so the first 2 years of life, you see that most of the oligosaccharides decline in concentration, with a few exceptions. And the exception here is 3-fucosyl-lactose, and the panel B here, that increases dramatically. And you see that 3-sial-lactose also increases, not as dramatically, but certainly to a certain extent. Everything else, like 6-SL, for example, over here, decreases within the first couple of months, and then kind of has a almost trace amounts left later time points. And we can take the same data here and show that with relative abundance. Here, teal color is the 3-fucosyl-lactose, and you see how that takes over the course of lactation. And that's quite interesting because we would love to know why that is. Is there any need for certain oligosaccharides to be more abundant at later time points? Does the baby need them for certain developmental milestones? We don't know that yet, but it's quite remarkable that in any study that we've looked at so far, whether it's here in North America or in Australia or in Europe or in South America, we see in longitudinal data that it's always the same oligosaccharides that increase over time. We would, of course, love to know why that is. Here's a slightly different way to look at oligosaccharide composition. This is a 3-dimensional principal component analysis where each dot in this 3-dimensional space here represents an entire HMO composition. So not just one oligosaccharide, but a composition. And you see the closer the dots in this 3-dimensional space, the more similar the oligosaccharide composition, and the further apart, the more different. And you can see that there's these 2 clusters in here, the left cluster and the right cluster. And these are our secretors and non-secretors. And careful with this one. So it doesn't mean that a non-secretor doesn't secrete any milk or doesn't secrete any oligosaccharides. It just means that they make very different oligosaccharides. So a secretor, for example, has very high levels of this oligosaccharide 2-fucosyl-lactose, whereas the non-secretors have had hardly any of it. And that is simply due to a single nucleotide polymorphism that switches off the transferase that puts fucose in this alpha-1,2 linkage. And I always find this absolutely fascinating that there is one single nucleotide polymorphism, one SNP, one base pair that's different between these different people. And it changes milk composition completely. Completely. So it's not just the presence or absence of this one oligosaccharide, it changes all of the oligosaccharide compositions because, of course, they're all connected in their synthesis. So think about that, one base pair different, and it makes a complete difference how your milk composition looks like. And that's different in different parts of the world. South America, we have almost exclusively secretors, and on the African continent, we have 35, in some areas, 40% of the people that are non-secretors. And of course, we would love to know why that is as well. Okay, so that's what we're trying to puzzle together right now. We use genome-wide association studies, transcriptomics, HMomics, and then try to puzzle this all together in silico and then target validate it in vitro to really understand how oligosaccharides are made in the mammary gland. And that's still a big unknown. And then we see, are there any other factors in addition to genetics that potentially help us modify composition of oligosaccharides? So can we, for example, recommend a certain diet, or can we tell mom to exercise in a certain way to change oligosaccharide composition in a certain way that might be more beneficial for the infant? So without going too much into detail here, that is a very active area of research to see if we find that specific oligosaccharides are beneficial, what can we do about it? What kind of recommendations can we give mom to do to achieve a better or different oligosaccharide composition? All right, so that's all for the maternal drivers at this point. There's a lot more data on that, but that's not the focus for this particular presentation. Let's talk about what these oligosaccharides actually do. What's their benefit for infant health and development? So basic mechanism, we think that these oligosaccharides are more than just That's what people always think. They're the prebiotics of human milk, and to a certain extent that might be true, but we think there's way more to it. And the story on necrotizing enterocolitis highlights that quite nicely. So one step back, what happens to the oligosaccharides when baby ingests them with the milk? Not just baby, actually, we've tried some oligosaccharides ourselves, and the same likely happens in adults as well. So oligosaccharides are very resistant to the low pH in the stomach, to pancreatic and brush-border enzymes. In the intestine, about 1% is absorbed intact. You can measure it in the urine. And then in the colon and more distal parts of the intestine, these oligosaccharides are either degraded by bacteria, so they're really prebiotics, or you can find 10 to 80% excreted in the feces. And this wide range of 10 to 80% depends on where you are during lactation. In very early phases where you don't have a microbial community established that can utilize these oligosaccharides, you find a higher degree of oligosaccharides excreted. And then once breastfeeding is fully established and the microbial communities are fully established, you find less oligosaccharides come out in the other end, so they're utilized more. And then towards weaning, that changes again, and you find other oligosaccharides being excreted as you introduce different foods as well. Your microbial communities adapt to that. Okay, so here in this diagram, you see the interactions between oligosaccharides depicted on the top, potential good bacteria in green, right on the left, potential pathogens in purple on the right, intestinal epithelial cells down on the bottom, and then underneath we have immune cells. And the notion is usually that oligosaccharides serve as prebiotics. So there's some bacteria that can utilize oligosaccharides. They have a growth advantage. They spit out some other molecules afterwards, like short-chain fatty acids and other metabolites. And then that becomes beneficial to the host, and that's the end of the story. But that's not the end of the story. So, for example, we've seen that some oligosaccharides have the opposite effects on some of the pathogens. They have antimicrobial effects and really keep the growth of certain bacteria in check. Group B streptococcus is a great example where we interview other labs have shown that you bring group B streptococcus growth to a halt if you add specific oligosaccharides. So that's quite a powerful way to look at this. We also see that many bacteria need to attach to the intestinal epithelial surface. They attach to glycans, to sugars, the glycocalyx of the epithelial cells. And that is usually required for these bacteria to hold on and proliferate and find their niche. But human milk oligosaccharides mimic some of those structures, some of those glycan structures on the epithelial cells, and they block adhesion by serving as soluble decoy receptors. And that's what we call the anti-adhesive effects of oligosaccharides. And that's shown over here, where you have a pathogen approaching the epithelial cells, and they try to attach to the glycocalyx to be able to proliferate and then eventually invade if you have an invasive pathogen, and that causes disease. Now, if you go one step back and look at the same animation here in the presence of human milk oligosaccharides that now mimic some of the structures of the glycans that we have on the epithelial cells, bacteria bind to the human milk oligosaccharides, and then their blocking mechanism or their adhesion mechanism is blocked, and they can no longer attach to epithelial cells and get washed out without causing disease. Okay, so that's the anti-adhesive effects, but then we've also shown that oligosaccharides have direct effects on epithelial cells independent of microbes, and sometimes that leads, for example, to of antimicrobial peptides and other things. So it can change microbial communities in the lumen, but without having a direct effect on them. It's more an indirect effect by changing the programming or excretion of epithelial cells. And then there's a direct effect on immune cells. We've seen this in macrophages, we've seen this in T cells, we've seen this in neutrophils, that they directly interact with immune cells and then have an effect. Again, that can backfire into the lumen and change microbial communities. But also, since about 1% of the oligosaccharides is absorbed, the concentration is still sufficient to have systemic effects as well. And we've seen this for macrophages, where IC50 for the effect of macrophages is around 10 to 15 microgram per ml, which is quite what we get in the systemic circulation, imagining that we start out with 10 to 15 grams per liter. And we think that this is probably just the tip. There's a lot of stuff to discover still, where we think that there might be direct effect of these oligosaccharides on, let's say, liver metabolism, muscle metabolism, and many other things that we haven't really discovered fully yet. Okay, so that's great. When is he starting to talk about necrotizing enterocolitis? I'll get there in about 3 minutes. But just bringing back these statistics here, I showed you in the beginning that we still lose about 2,200 children under the age of 5 to diarrheal disease every single day. And we see that individual oligosaccharides contribute to the benefits of human milk. For example, 2-fucosyllactose working on Campylobacter and reducing Campi infections. LNT having effect on group B streptococcus, I mentioned that earlier. But we've also seen this for the protozoan parasite Entamoeba histolytica. And then 3SL, 6SL working on certain rotaviruses. There's new data that comes out for norovirus, for some other viruses, potentially including COVID. So we have a lot of different oligosaccharides here, and that's probably why we have so many that can interact with many different pathogens and many different adhesion molecules on pathogens to really block their effects, being either anti-adhesive or antimicrobial. We see very strong correlations between human milk oligosaccharides in mom's milk and infant growth. And this can go at two sides of the growth spectrum. This can be for growth that leads potentially to overweight. And this is 2FL being associated with growth in infancy and early childhood, but also body composition measures. And interestingly, the opposite is true for neo-LNT. Those two oligosaccharides, the ratio of them seems to be more important than the concentrations of individual ones. And we've seen this now in multiple different cohorts in the US, in Denmark, in Finland, and most recently in a manuscript that just got accepted 2 days ago in Australia. So it's always the same oligosaccharides that pop up that are associated with growth measures, with body composition measures in the Western world. And then Jeff Gordon and others have done some work where they looked at malnutrition, and they identified other oligosaccharides like 3SL and 6SL that are associated with preventing malnutrition. So you see, again, different oligosaccharides work structurally dependent on very different aspects of the similar story, but on different aspects of it, either overnutrition or malnutrition. And then for immune development, going through this real quickly here, different oligosaccharides being associated with cytokine release, macrophage resolution, and other things. But here we find more in a real world scenario that it's not individual oligosaccharides that are associated with certain outcomes, but it's a mix of different oligosaccharides. So ratios matter of different oligosaccharides. We see this for cow's milk allergy, food sensitization, allergic disease. So when it comes to somehow priming immune systems or immune responses, it seems to be more a mixture of oligosaccharides instead of being one single structure or one single epitope. And skipping this here, that makes sense because the immune system is not just one cell type. It's not just one receptor, because any immune cells here has different receptors, and many of those receptors interact with different glycans. So we know there's cyclics, galactins, selectins. They all interact with glycans, and they interact with different types of glycans. So different human milk oligosaccharides, if they interact with immune cells, have the capacity here to trigger immune responses in a certain direction. And it's the concerted interaction of different oligosaccharides working on different receptors of different immune cells that really work in concert and make the music in the end. Okay. And then one more thing here before we get to NEC is on neurodevelopment and cognition. Previous few years, we've seen that silated oligosaccharides had been associated with improved neurodevelopment and cognition in either animal models, mostly animal models. But most recently, we've really shown that the oligosaccharide 2-fucosyllactose, both in animal models as well as in human correlation studies, seem to be somehow involved in improved cognitive development. And that's quite interesting because this particular oligosaccharide is available at large scale now to be deployed and be used. And this could potentially be not just in the infant space, but also then in the other end of the age spectrum where cognitive development is in decline, and oligosaccharides might potentially help. Okay, but now finally, halfway through the talk, let's talk about necrotizing enterocolitis. Okay, so I showed you this statistic here before that we still have about 5% of all very low birth weight infants developing necrotizing enterocolitis. and often deadly intestinal disorder. So NEC remains to be one of the most common and devastating intestinal disorders in preterm infants. The mean prevalence in infants with a birth weight under 1,500 grams is about 5 to 10%, with very high mortality rates. Depending on severity, this can go between 50 and 100% mortality. And the survivors are often faced with long-term neurological complications. So it's a very severe and not infrequent disease, not uncommon disease in preterm infants. And the difficult part is that it comes on rapidly. So you can see a baby at night at your last visit, and then overnight develops NEC, and the morning the baby is in very devastating condition. And we don't really know what triggers that rapid onset and what we can do about it. So etiology and pathogenesis is likely multifactorial and poorly understood, which doesn't help. And the rapid onset and fulminant progression and limited treatment options really make it most desirable to prevent NEC altogether before it strikes, because once we have NEC in place, it's extremely difficult to manage and often has a fatal outcome. Okay, but then we have human milk, and I showed you this before as well in the introduction. We know that preterm infants who receive human milk are at a 6 to 10-fold lower risk to develop NEC. So providing human milk in the NICU is critical. And unfortunately, we still see this way too often, that human milk is not part of the plan, the management plan for the preterm infant, and it should be, because there's clear data. If for anything in human milk, the data is extremely clear for necrotizing enterocolitis, that if you feed human milk, you're at much lower risk. If you introduce infant formula, your risk goes the other way. So I would say it's almost mandatory that our babies in the NICUs, preterm babies, receive human milk. Data is extremely strong. The question is, what is in human milk that protects the preterm infant from necrotizing enterocolitis? Is it human milk oligosaccharides, or do human milk oligosaccharides at least play a role in this protection? And why do I say that? Well, simply, human milk contains a lot of human milk oligosaccharides, whereas infant formula, at least in the days when we started this hypothesis and this work, did not contain any oligosaccharides, or very, very little from the raw materials that were used to make infant formula. So that was our hypothesis when we set out about 12 years ago to test whether human milk oligosaccharides contribute to the beneficial effects of human milk when it comes to preventing necrotizing enterocolitis. And I'm going to develop this diagram here with filling in data that we've produced over the last 12 years. And you see this is reviewed and highlighted and all combined in a review article that we published a few years ago. Okay, so what we did first is we isolated human milk oligosaccharides from pooled human donor milk. So, and we did this from pooled milk to make sure that we get the garden variety of all oligosaccharides known, and that might be different from donor to donor. And then we tested these oligosaccharides that we isolated from milk in an animal model of necrotizing enterocolitis, where we use rat pups here that we feed ourselves now with either formula or formula that contains human milk oligosaccharides. And you see we feed them here ourselves. You can see the success of the feeding, where the belly here pops up to be yellowish white. That's where the formula went in, so you know you had a successful feeding. You remove the feeding tube, and then there's the next guy waiting in the background. You can do this with about 150 different animals at a time, and then you would start over again with the next round of feeding once you're done with that one. So it's like having a baby at home every 2-3 hours, you can start feeding again. Okay, so we did that, and to our surprise, the data was extremely strong, just looking at survival due to NEC-related complications. So you see survival plot here. It starts at 70%, just to not mislead you. And we see that in the formula-fed group, we lost about a quarter of the animals after 4 days to NEC-like symptoms. If we leave the animal with mom and just expose it to hypoxia, just like we do with all the other pups, but the difference is that these are fed by mom and not by us. You see that we lose none of the animals. Not surprising, none of the animals develop necrotizing enterocolitis. So we lose 25% in the formula-fed group, none in the dam-fed group. If we add human milk oligosaccharides isolated from human milk, the survival goes up to almost where the dam-fed group is. We only lost one of the animals to NEC-like symptoms here. And that to us was striking. Like, we did not expect that dramatic effect. And then we looked, of course, at the intestines. So we dissected the intestines. You can already see the intestine here that is from the formula-fed group is much darker, some more blood in the tissues here. We then did ileum sections, stained with H&E, had a pathologist look at these blinded, and then scored. And these are the scores from 0 being healthy to 4 being a completely destroyed intestine, really. And we find in the dam-fed group, so that's the animals that stayed with the mom for feeding, most of these intestines are healthy. There's no big issue with that. However, if we feed them ourselves with infant formula alone, you see that there's a huge variation in pathology scores with an average around 2. Some of these intestines were completely destroyed, and a couple were still remaining healthy. Now, same formula, same feeding protocol. The only difference is that we added human milk oligosaccharides in this third group. Pathology scores dropped significantly, almost down to the dam-fed control, which matches nicely the survival data. Now, if we add a control oligosaccharide, galacto-oligosaccharide, that is not in human milk, that is an artificial galactose polymer, you see that that didn't have an effect. So the pathology scores are still somewhere around 2. So it's specific to human milk oligosaccharides. Not any oligosaccharide can do it. But then let's not forget, there's about 150 different oligosaccharides, and we isolate them from pooled donor human milk. So we have the entire set of oligosaccharides in this fraction here. The question is now, is it all human milk oligosaccharides, or is it one specific one? So to answer that question is we used a multidimensional chromatography approach where we use the structural features that are known to human milk oligosaccharides, their charge, first of all, so how many sialic acid, how many negative charges are on the oligosaccharide. And we separated the oligosaccharides accordingly, and we found that the most effective fraction of oligosaccharides was the one that contained 2 sialic acids, had 2 negative charges. And then we looked at the structures. There's more than one oligosaccharide that contains 2 sialic acids. We did mass spec on that to confirm that that's really the case. And then used the second dimension. We used the size chromatography, where we now say, okay, we have oligosaccharides with 2 sialic acids, but they can be either very small or the backbone can be large, and separated the oligosaccharides accordingly. And here we found one oligosaccharide, and that's what you see here in the size exclusion, this biggest peak here, oligosaccharide 2, that had the most significant effect. So now we have one oligosaccharide that has the biggest effect for the NEC reducing or pathology score reducing abilities here. And the question is now, what is this oligosaccharide, right? So you can't just take a look under the microscope and see like, oh, this is how the oligosaccharide looks like. That's not possible, of course. These are very small molecules. So we had to do some extensive analysis here, glycobiology analysis or glycostructure analysis, first with sequential exoglycosidase digestion, and then did linkage analysis by GCMS, and puzzled together how this oligosaccharide might look like. And this took us about 9 months back then, just to elucidate the structure. And we found that this was the active structure. It was called DSLNT, which is short for disilylacto-N tetraose. Okay, so we did that, did some chemical space analysis around that. But then the question was always, well, how good is your animal model? And can you show us that this actually works in humans? Well, by that time, DSLNT was not available in concentrations and costs to really run an intervention study. It would have been extremely expensive. So we did an intermediate step and conceived a human cohort study where we recruited mothers and their very low birth weight infants, collected milk samples every second day, milk that was fed to these infants every second day, analyzed oligosaccharide composition, and then linked that to the clinical metadata to see which infant developed NEC and which infant didn't. So hypothesis here was that infants who develop NEC receive milk with less DSLNT than infants who do not develop NEC. And we're only looking at the human milk-fed infants at this point. Study was done in multiple different sites in North America with San Diego as the prime. And this is an example of what kind of samples and data we collected. This is representing the first 28 days of an infant that developed NEC. And you see here in day 16 to 22, that's where the orange boxes are that says N on it. That's when the infant had NEC. We had milk samples collected on day 8, 10, 12, and 14. That's where the numbers are. And then after NEC, we had milk samples collected on day 23, 25, 27, and 28. And then we analyzed the oligosaccharides on that and matched that with infants that had similar birth weight, similar gestational age, similar delivery mode, similar race, and where we had samples at about similar times. So we were able to compare oligosaccharide composition. And what you see down here is the first 28 days postpartum on the x-axis. And on the y-axis, you see oligosaccharide concentrations here, the sum of all oligosaccharides. The gray dots in the background are our controls. So these infants did not develop NEC. And then yellow is the Bell stage 2, red is Bell stage 3 NEC. So those are the infants or milk samples that represent oligosaccharide composition with the milk that went into those infants. And you see there is no significant difference. So no difference in the total amount of oligosaccharides that go into an infant that either develops NEC or doesn't. But then that wasn't the hypothesis. The hypothesis was that one specific oligosaccharide, namely DSLNT, was lower in the infants that later developed NEC. And that was the case. And it was very striking. So you see here, just for DSLNT, every single child that developed NEC in the end received human milk with lower concentrations of DSLNT. And remember, DSLNT was the oligosaccharide that was protective in the animal model. And here's a different way to showcase that every box that you see here on the time box scale here that has a color in it is a milk sample that we analyzed, and the color represents the average or represents the oligosaccharide concentration over average. And the more red the color, the lower the concentration of this oligosaccharide in that milk sample. And you see for DSLNT, for all the NEC cases, the DSLNT concentrations are below average compared to the controls. That was not the case for any of the other oligosaccharides. So highly specific to DSLNT. You can calculate the odds ratios, put this all together in a model and see how significant this is. You can then see, well, can we develop decision thresholds where we say, well, if we find a milk sample where the oligosaccharide concentration is above or below this certain threshold, how likely is it that an infant developed NEC? So that will be helpful to screen mom's own milk and also screen donor milk and other human milk products for potentially insufficient concentrations of this oligosaccharide that is protective. So this was one study. We've since seen this in two other independent studies, one in South Africa and one here that I'll show you now in the UK, where we teamed up with Chris Stewart up in Newcastle. They have a large biorepository of milk samples from NEC infants and respective controls. So we ran those milk samples and we found that there is indeed one oligosaccharide that stood out to be different between controls and NEC, and that was once again DSLNT. Highly significant, just like in the previous study that we had done ourselves. And you see that even their structural concentrations are very similar to what we had found in our North American cohort. So it's around 210 to 250 microgram per ml. So that shows that there is a match, that we have that the cohort studies, 3 independent cohort studies, really match what we found in the clinical in the preclinical animal model, and that now gives us greater confidence to move forward with a clinical trial. So what we can say from that is that we've shown that HMOs are somewhat at least contributing to the beneficial effects of human milk and preventing NEC. And it's not just all oligosaccharides, it's DSLNT in particular that has the biggest effect, and that was shown to be both protective in the animal model as well as associated in the independent cohorts. And that's summarized here. And really that gives us now an opportunity to develop therapeutics. So how would this look like? And that brings us to the last section here on therapeutics. So can we make DSLNT available at larger scale, low cost, feed it to all of our preterm babies to prevent NEC? We could do this either by chemical total synthesis, extremely expensive. You get a few microgram, and that certainly wouldn't be sufficient. You could do a mixture by adding chemoenzymatic synthesis, Also still very expensive, and you need all the different precursors for that. It needs lots of enzymes, certainly also not feasible at scale. Then there is synthesis in bioengineered microbes, and that's what's currently happening for some of the smaller oligosaccharides, like the 2-FLs, the 3-SLs. We can produce those in large fermentation tanks, and that's what's currently added actually to infant formula that you find on the shelf that says it contains HMOs. It really contains a couple of oligosaccharides that are like the tri- and tetrasaccharides. But the larger molecule here, DSLNT, is not feasible to be produced in these fermenters yet, and it doesn't look like that's coming anytime soon because of its complexity. Right now, we did the calculations. If you wanted to do that, it would cost you about $900,000 to make DSLNT available to preterm infants, and that's per baby. That's for the first 28 days of life. It would cost you about $900,000, almost $1 million to put DSLNT into the feeding plan. So it's certainly not feasible. However, there's another opportunity, and the opportunity is to screen mom's own milk or donor milk products for potentially sufficient or insufficient levels of this oligosaccharide. So we could do that in the NICU directly. If mom is producing milk, we would screen the milk and say, well, DSLNT levels are great, feed it to the baby, no problem. We can do this in the milk bank, where we would specifically screen for DSLNT high concentrations and then use those batches of donor milk that have high DSLNT levels and feed that to babies in the NICU. The problem there is, or let's go one step back here real quick. How common is that actually? So we looked at the distribution of DSLNT concentration in over 200 active qualified milk donors in the US. And this is your distribution of DSLNT here in the different samples. And if we now take a look at where is the NEC preventing threshold of about 210 microgram per ml in North America, that would be right here. So that means that there is about 23% of the milk samples of active donors that would be below that NEC preventing threshold. So to have an opportunity to screen would make a huge difference. The major impediment here is, though, that there is a lack of technology to measure DSLNT or human milk oligosaccharides in general at point of care. So we currently don't have any technology to just simply go in, put a drop of milk on a stick or something, and then say, oh, DSLNT concentrations are X, Y, Z. So usually there's only a few laboratories in the world really that have the ability to measure human milk oligosaccharides. It takes a lot of expertise and expensive equipment. And the time from sample collection to analysis, if you even wanted to do that, is several days at least, which is certainly not useful given NEC's rapid onset and fulminant progression. By the time you have the data back, your baby might be dead already. Okay, so for us, innovation lives at the intersection of disciplines. So we took to our engineering department here and teamed up with Dr. Drew Hall, who is in the Department of Electrical and Computer Engineering, and one of our 2020 Mommy Seeds Pilot Grant awardees, who does exactly that. So you see him here in the lab, standing with this device in his hand, which he's been using for multiple different biomedical applications. It's a flow device where you can put in a certain amount of liquid, and this could be urine, this could be milk, this could be other things, to then do some magic and measure certain components at point of care. And this is how this looks like from the inside, where you have this readout, this thermal control with Bluetooth communication, where you can then send data over to your Bluetooth enabled cell phone to then see, okay, here's the data, your DSLNT concentrations are this and this high. This is good to feed, or maybe use donor milk that's high in this specific oligosaccharide. And we've seen this before. So we had a proof of concept paper that we published last year. This is not for DSLNT. This was specific for 2-fucosylactose, where he managed to build a device that can measure that with high accuracy. And for DSLNT, we're now moving to a slightly different technology without going too much into detail, using molecular imprinted polymer technology and then electrochemical analysis coupled with that to detect DSLNT. We've seen this actually works with human milk and with DSLNT, so we're testing this out already. And then planning on using surface imprinting to make it more specific to DSLNT, since there's lots of other oligosaccharides in human milk. And that's where we are right now, where we're testing for sensitivity, specificity, making this more robust, and then teaming up with our leaders in the NICU and in the milk bank to develop this handheld reader with a focus on usability and robustness to prevent NEC by looking at DSLNT concentrations in human milk. Okay, so the idea is we can go into the NICU, we can go into milk banks, deploy this device, measure where we are with DSLNT concentrations in mom's own milk or in donor milk, and then prevent necrotizing enterocolitis by only feeding human milk or donor milk that is with sufficiently high DSLNT concentrations, and turn this NEC baby hopefully into a happy baby that can leave the unit and live a healthy life without any major complications. Okay, already way advanced in time here. I want to leave some time for questions as well. Just a quick summary. HMOs are the third most abundant component of human milk. Composition varies greatly between individuals. They're just mostly driven by genetics, but can change to a certain extent by other factors. HMO composition in the same individual is remarkably constant over a short period of time, but changes over a longer period of time, with some oligosaccharides increasing, most of them decreasing. We don't really know why that is, but would love to understand why. HMOs remain undigested by the host and reach distal parts of the GI tract, where they can serve as prebiotics, but they do many other things as well. So we don't want to have a microbiome or a probiotic that utilizes up all of our oligosaccharides, because we want them to do a bunch of other things, namely serving as antimicrobials, anti-adhesives, epithelial cell modulators, immune cell modulators, and many other things that we haven't fully understood yet. We also know that HMOs are absorbed intact from the gut, reach the systemic circulation, and they can have a bunch of other effects after absorption. There's accumulating evidence that indicates that HMOs may be associated with infectious diseases, lower risk of infectious diseases, that is, improved immunity, better cognitive outcomes, and then a lower risk for preterm infants to develop NEC. And that's where the focus of this presentation was on. Necrotizing enterocolitis is associated with low human milk concentrations of a specific HMO called DSLNT. And we've shown this in multiple independent human cohort studies, as well as significantly reduced with DSLNT interventions in an animal model. And we've seen this in a separate animal model in the meantime, which opens opportunities to develop new therapeutics, which are unfortunately currently not available due to high costs and low scalability. We're hoping to get there eventually. But a more elegant approach here to stay in the human milk field and keep on feeding human milk is by developing new technologies to screen human milk for DSLNT at point of care. So that means in the NICU and milk banks, to make sure that we only feed human milk that is high enough in DSLNT concentration. Okay, I leave you with this quick invitation. We have, like I said, a new Human Milk Institute at UC San Diego. The official launch will be March 30. Registration is free of charge. And we have a few other things going on at the same time. So I invite you to come to that if you happen to be in San Diego or would like to come to San Diego. Not a bad place at the end of March. And then in addition, we have a conference on the origins and benefits of biologically active components in human milk. Lots of HMO, lots of other milk components being discussed there. And that's going to be in June in Melbourne, Florida. Okay, I'll leave you with that and hope there is a little bit of time for question. I stop sharing so I can actually see who's on the call. Thank you.
Thank you, Lars. Do we have any questions from the audience? If you so, please raise your hand or type it in the Q&A box.
I see one question in the Q&A box here while other people are thinking. And the question is, any effects of drugs on human milk properties and quality? Great question. And that's exactly what our MPRINT CET is set out to do, is to look not only whether drugs are transmitted through human milk from mom to baby, but really if maternal drugs change milk composition. So this could be volume, this could be molecular composition. And we have a few examples where, for example, of course, for the vaccine, for the COVID vaccine, we have a high interest in studying that. We see that there's some changes on volume, but there's certainly changes on molecules like oligosaccharides, some immune factors. Of course, the immune globulins go up in response to the vaccine. That's the whole purpose of it. So yeah, that's a very active field of study right now, how human milk oligosaccharides or how human milk composition is changed by exposure my maternal exposure to certain drugs.
So there's another question in the chat. Can you speak more to the rat experiment and specifically milk given versus by lactation, thinking of the implications for pumped milk from the mom?
Yeah, so we did feed the pups ourselves with formula. We did not feed the pups with pumped rat milk. Extremely difficult to milk a rat, and this is not a joke. It's much easier to milk a small mouse than to milk a rat. I don't know exactly why, but rats have a very different tissue organization when it comes to the mammary gland, makes it very difficult. I can't tell you really from the rat experiment whether pumped milk or fresh milk is more protective. We know from the oligosaccharide world that whether you pump or whether you pasteurize milk, it doesn't affect oligosaccharides as much as it does many other milk components. So that I would maybe leave as a vague answer without having the full knowledge and answer.
I think there's a couple of questions talking about the variability of HMOs over time or with other covariates, for instance, maybe effect of maternal nutrition, or whether maybe at a certain point in lactation, if they're higher than at other points, or, the other question we have is whether it's, a mom who's exclusively pumping would have different HMOs than one who's also breastfeeding.
Yeah, that's a huge area of research right now. And I touched a little bit on that in the beginning of the talk and kept it quite short since I want to get to the neck part. So we know that genetics certainly is the biggest driver of human milk oligosaccharide composition. And there's a few things you just can't do anything about. Either you are, a secretor or non-secretor, you can't change that. You cannot change that with nutrition or anything else you can do to mom. But there is other subtle or more subtle differences where we see that nutrition, either in animal models or in humans, does change oligosaccharide composition. We see that physical activity has a certain impact on oligosaccharide composition. We see that giving a probiotic to moms during pregnancy can change oligosaccharide composition afterwards. So there's a few things we can do, still a lot to learn, but more important right now is, other than DSLNT for necrotizing enterocolitis, we don't really know what the optimum oligosaccharide composition is. So it's great to know that you can change it, but to what do we actually want to change it to? And for DSLNT, not much known about what could potentially affect that. We didn't see a lot of changes in DSLNT in the nutrition studies or in the physical activity studies. It's more the smaller oligosaccharides like 3SL. So currently, we don't have anything where we can say, hey, mom, do this, that increases your DSLNT concentration.
All right. Hopefully someday we'll find that information and maybe have another therapeutic that you could give to mom that would then impact the baby. So
that would be ideal.
Great. I don't see any additional questions, and we are at the top of the hour. So I will thank you for an excellent presentation, and we look forward to seeing everyone at our next webinar. Thanks.
All right. Thank you.